Welcome to the first annual Symposium on Mitochondrial Health, Healthspan, and Aging. Now, a panel discussion moderated by Rob Lustig.
So, I don't know about you, but that was a heady day, and I think you all. And I think you all learned something. And what I'd like to do is focus on, to start with, is focus on the common themes that we heard out of today. Okay? Stress. Sorry?
Stress. What kind of stress?
Stress. Yeah, we can talk about stress. We're going to talk about mitochondrial dysfunction. So, the question, of course, is, what comes first? How do the external stimuli ultimately impact on the internal milieu of the mitochondria to cause these things, and why does the mitochondria respond in the way it does to try to deal with this? Why did the mitochondria become sort of the arbiter of this metabolic mayhem? Bob, this is kind of, from a teleologic standpoint, evolutionary standpoint, kind of your baby.
Well, I mean, I think mitochondria...
And thank you, Dr. Mattson, for joining us.
Yeah. Mitochondria and, the original archaebacteria, that began that, that took part, well, the proteobacteria and archaebacteria that took part in that original contract 2 billion years ago, have been working out their differences, for 2 billion years. And it was truly a symbiotic relationship that ended up actually being necessary for all cell differentiation in metazoans. So the ability of mitochondria to adapt to local conditions allowed, different cell types to perform different functions, as they developed. So anyway, it is an ancient contract, and there was a division of labor. There's a set of genes in the nucleus, but then also mitochondria were made the sentinels of the cell to constantly monitor, because the rest of the cell really is receiving information in terms of chemical metabolites from mitochondria about the safety and danger of that cell. And so I don't know what to but it's just, yeah, I'll leave it at that.
One of the things that, as an endocrinologist, has always sort of floored me is, you were talking about division of labor, steroidogenesis.
Sure.
So steroids, start as cholesterol and get converted to pregnenolone in the mitochondria. Then pregnenolone leaves the mitochondria to go into the cytosol and then gets converted to whatever the hell it's going to be converted into. And then 11-beta-deoxycortisol comes back into the mitochondria to be converted to cortisol, the ultimate stress hormone. I have never, for the life of me, understood why such a circuitous route, but there must be some teleologic explanation for why, our mitochondria do that, and what role mitochondrial dysfunction plays in that stress response. Any thoughts on that, either of you?
Sure. Well, I can just say, I mean, what better location to decide on the activation or inactivation of cortisol than the molecule that is, or the organelle that is tasked with the ability to sense environmental stress and danger. So. To me, it seems logical, that transition between activating and inactivating cortisol depends on, aspects of mitochondrial metabolism.
Any thoughts?
I just want to follow up on the first point before we move on to that. Essentially, what we're talking about really is physiology 101. So if you think about Hand-Salier stress theory, or preconditioning of the heart. So if you were to occlude a heart acutely and get X amount of damage, but if you give it a little bit of ischemia and then you occlude it by the same magnitude, there's much less damage. If you plunge into cold pools and you start getting your body used to it, your body will adapt. So there's so many different danger responses that can give you a similar outcome. Exercise, we have heat going up, we have ATP going down. There's so many different stressors that are going on. We respond to it, and that's why exercise training, if you went out and tried to run a marathon, you know, the first time nobody could do it. But eventually your body gradually adapts to all of these things. So really, it makes sense that the recovery from all of those adaptive responses that we have to respond to or we will die needs a very energy provisional organism, which is clearly the mitochondria, which is why it really comes down to so many things. For example, going into a sauna, for example, people are even looking at that to induce heat shock proteins, and different heat shock proteins like HSP72 can upregulate the mitochondria. So there's many ways to get to the same end, and ultimately it comes down to, you know, basic physiology that you learn in first-year biochemistry class.
I'm going to take it in a different direction, and actually, Dr. Mattson, I would love for you to weigh in on this thought. As you know, my second favorite hobby horse, it used to be my favorite, but now it's my second favorite, is fructose, right? My favorite's now mitochondria. Okay, but my second favorite. In the brain, you know, people always said, well, fructose isn't a substrate for neuronal metabolism, and that is actually true. It is a substrate for astrocyte and microglial metabolism, but not neuronal metabolism. So the neuron does not metabolize fructose. It doesn't have the enzymatic machinery to do so, but the astrocytes and the microglia do. As we heard, the microglia actually are the one place in the brain that has the GLUT5 transporter to be able to transport fructose. Now, obviously, your brain's not supposed to be exposed to a whole lot of it. That's why you've got this first-pass liver effect, except when you drink, a soda, 12 ounces, 140 calories, you're going to get a fructose response. You get a 6 micromolar fructose response. That's going to go to your brain. And the question is, what does it do there? So we heard a little bit today about the fact that 90% of the synapses in the cortex are glutamatergic, okay? And 10 or 15% of them are GABAergic. But what was left out of that discussion was the fact that glutamate is the precursor to GABA through the enzyme glutamine synthase. So glutamate, okay, in the presynaptic neuron becomes glutamine within the astrocyte, which then gets exported back out, gets converted back to glutamate in the GABAergic neuron, and then goes to GABA. So there are 3 separate enzymes necessary to get from glutamate to GABA in 3 different neuronal structures: the glutamatergic neuron, the astrocyte, and the GABAergic neuron. Now, how dumb is that? Okay, it's a bizarre cycle. Why do I bring all this up? Because fructose inhibits glutamine synthase directly. And the question is, sorry? Oh, Mark? I can't hear you. Wait. Mark, hold on. We need sound. There's no sound.
Yeah.
Okay, we have sound now.
Can you hear me?
Yes, now we can.
I think it makes a lot of sense, actually, because you're essentially recycling the same molecules with enzymes, and you're not having to produce them, at a distance and move them there. So I think this, the glutamine then being converted to glutamate and then to GABA makes a lot of sense to me. And also the astrocytes are typically physically closely opposed to the synapses. They kind of wrap around the synapses. So it's not like the astrocyte releases glutamine over here and then it diffuses over here. It's like right there.
Right.
So it makes a lot of sense to me.
Well, except for the fact that since glutamate is excitotoxic and GABA is inhibitory and actually puts the neuron to rest, you'd think you'd want a whole lot more GABA than glutamate, but it's the opposite way, and you've got a way to stop it.
But the thing is that there is very little glutamate outside of cells at all. It's very active. As soon as it gets outside of the neuron, it's actively transported into cells by a glutamate transporter. So there's high levels of glutamate inside of neurons, but it's not activating receptors. And to get excitotoxicity, you have to activate receptors.
Okay.
And it is actually a normal strategy in metabolism to, if a molecule starts out as an excitatory molecule like GABA, that its natural metabolism leads to an inhibitor that then closes, that completes the cycle of excitation and relaxation or inhibition. Excitation and inhibition. So even ATP signaling is similar, where ATP is excitatory, but it's metabolized to adenosine, which is inhibitory.
Yeah, that's a good point.
So actually, that brings up a question I had outside. If that's the case, and if ATP is being dumped into the bloodstream, and you can measure it there, how about a drug or a nutraceutical that actually cleaves off those 3 phosphates, turns it into adenosine, and gives you some deep rest?
Yeah, interesting that you should say that, because in experimental mice, it's actually possible to use a bacterial enzyme called apyrase that does exactly that. That will cleave ATP down to adenosine and close the cycle. It can't be used long term, because the animals make antibodies to it, but it's a short term solution. Anyway, so that's it. So it is a strategy.
Is there a small molecule pathway for that?
Well, it turns out that cells have ectonucleotidases on their surface. So CD73 and CD39 are ectonucleotidases whose job it is to cleave off those phosphates in order to convert ATP to adenosine.
But you wouldn't want that to cross the plasma membrane and do that inside the cell.
Right.
You have to stay extracellular.
Yeah.
Right, exactly.
Right, it would have to stay extracellular, or you'd end up being paralyzed. Yeah.
That's called rigor mortis.
Right. Fair enough.
So which was actually the part, Bob, in terms of extracellular ATP, how does it come out of the cell? Is there an active transport, but to the...
Well, so remember, inside the cell, the concentration is anywhere between 5 millimolar to 20 millimolar is ATP.
But going to the cell membrane,
very
polar molecule.
It's very tightly regulated. This is not a random process. It's not just because cells break open. There's a transporter called the pannexin 1, transporter that is actually very tightly regulated in how it opens and closes under stress conditions. And so under a variety of different stress conditions, and they range from redox stress to non-redox chemical stress to peroxidation of membrane lipids to cholesterol and ceramide accumulation around the transporter. That opens the channel and releases ATP and UTP, a number of different molecules that are large nucleotides. So it's very carefully regulated. It is
right. No, I can imagine. I'm just trying to figure out how does it go through the transmembrane region. Is it?
Well, it's not transmembrane. There's a channel. This is a channel.
Right, but it's now reverse transport. Is it just simply concentration gradient, right?
Well, it's a huge so there's around every cell there's a halo that we call the unstirred water layer.
Right.
that unstirred water layer can be about 50 nanometers in thickness. If you imagine it like a Costa Rican rainforest, if you were an angel above the canopy, okay, you'd be feeling the wind blow you back and forth. But if you descended into the canopy of the rainforest, you would then begin to smell the smell of the forest. Okay, it's this zone of the unstirred water layer which is the place where all ligand-receptor interaction occurs in a cell.
So I mean,
and it's much different than just the free water layer outside circulating in the blood.
Yeah. So I mean, so for example, you have unstirred water layer at the enterocyte intestinal coating too. Right?
At the what?
In the gut, right?
Sure, it's actually elaborated. Yeah. The glycocalyx is, it can be 100.
But there, still the ionic molecules go through intercellular space, tight junction, rather than transcellular, right? So there's, so they go through it. That's what I'm trying to see, that within the cell, you don't have any intercellular cell membrane. But that's what I'm trying to, so there's our...
that's another group of channels called the connexins.
Yeah.
Go ahead.
Mark. Well, this is what I was going to say. So, the connexin is kind of like a hemi-junction, I guess. So you can have gap junctions, which big molecules can pass through between cells. Like the cardiac myocytes are connected by gap junctions. That's why you get such fast, like waves of calcium-mediated contraction. And these connexins are they're kind of like a connexin hemi-channel. It's just half of it, but things can come out of a cell.
Yeah. So they're both connections intercellular and at the
and it's concentration driven. So from that point, is it possible to look at plasma levels of ATP?
Yeah. So that's what I was going to get back to. It's kind of the question that Rob asked.
To determine the degree of mitochondrial dysfunction or depletion as a
So the gradient goes, so the KD for receptor binding of different, of ATP and ADP is for different purinergic receptors is about 5 micromolar, which is 1,000 times less than the intracellular concentration. So you can imagine so that's about 5 micromolar. If you just go a few, 100 microns farther into the interstitial space, you get down to about 50 nanomolar ATP, and it's and in blood that's the level, unless you're really rough with how you treat the cells. Okay, it is 20 to 50 nanomolar, and it does not correlate with that stirred water layer concentration. So you have to have another way of interrogating the system, and we believe that one way to do that is actually to measure the expression, the receptor count per cell of different purinergic receptors on different cell types, because they will rise and fall according to the physiologic state of the cell. Yeah. So ATP alone would have been a great, target, but it is so potent physiologically. So I can say, if we do these experiments in mice, if you inject systemic ATP in an intraperitoneal injection into a mouse, within 15 minutes, it inhibits mitochondrial function systemically and decreases mitochondrial oxygen consumption by 74% in clams cages, in these metabolic cages. And it takes about an hour to 2 hours for it to be metabolized in the animals to recover. But in addition, it releases cortisol. I mean, so it's a direct, ATP is a direct, stimulus of cortisol release. And there's IL-6 and IL-10 actually production. We haven't measured IL-15. That was an omission. I wish we could write, but but anyway, the point is that single archetypal danger signal triggers many different cell types to take up the call and send systemic messages of danger and threat.
Let me go ahead, Mark.
There's just an interesting anecdote. I was sitting at a very similar discussion as this about 40 years ago, and athletes were apparently taking ATP and infusing it, and they were saying that at the end of the Tour de France, it was giving them the extra boost so they could go over the finish line. The point is, some of the crazy things that we as scientists trickles down to these athletes, and that was one of them. I mean, it's the worst thing you probably could do is infuse ATP. But crazy things like, we mentioned interleukin. So I was at another meeting where people were taking interleukins and injecting them into their muscle so they get the little bump so that they would have good definition in their arm. $25,000 for the interleukins that they were injecting. And now there's crazy stuff like people,
I got the cream and the clear for them.
But crazy stuff now, I mean, people are seriously doing gene therapy, apparently, supposedly with CRISPR, to alter their PGC1 alpha. The stuff that these athletes do is absolutely incredible. And it gets trickled down from misunderstanding of fundamental biology. And that's why it's important to have these discussions so that we can quell this misinformation.
So, I mean, it's important. The amount of ATP your body makes every day just to go through 2,000 calories is 70 kilograms.
Right.
So your body is making the equivalent of your body weight in ATP and turning that over every single day. So that you can imagine just taking oral ATP is nothing. It does nothing. You know, it's really all about what your cells are doing and how the cells are communicating with the brain in order to regulate.
I think that's one of the big take-homes from this. Is, you know, you're not going to make a whole lot of difference in terms of this kind of energy production and/or utilization.
But it's as crazy as the NAD craze. NAR, NMN, et cetera.
Yeah.
I mean, it's the same analogy. Eating a couple of grams even of this stuff, of course, all the studies showed that it didn't do a darn thing because you're making kilos of ATP, and then to provide a few grams even of NAD is just
what works in the dish does not work in the person. That's
but many people still swear by it. But that's
well, it doesn't matter if people swear by it. The randomized double-blind studies show absolutely no effect.
And including Mark, one of the people we know together, way back in the Citrus days and all, who's still promoting NMN.
Yeah, but they can promote it all they want, but the studies clearly show it doesn't work.
Let's switch gears. So, I want to get Elissa involved. Let's talk about the elephant in the room that we didn't talk about at all today, but it is, shall we say, directly related to this issue, and that's prenatal and neonatal and even childhood stress, adverse childhood experiences. Clearly, epigenetic effects, changes in methylation patterns. How does this affect the mitochondria? How does it affect mitochondrial metabolism? And how do you undo that phenomenon in order to be able to get to healing when things are, shall we say, so screwed up?
Okay, I'll take that one. Thank you. That's a low ball or a softball, just curious with a time check. How many of you have a question you would love answered?
Come to the microphone.
Okay.
I can't tell if you have a question unless you go to the microphone.
Yeah. So I'll go ahead. Is this about child?
No. Oh. This is in relation to the previous discussion that was happening. We were talking about NAD or ATP. It's not going to make any difference. But what do you think of the recent therapeutics on MRT? Like people are, it's even in clinical stages. People are infusing mitochondria. Of course, it's even in clinical setting. How do you think that could influence or otherwise? Yeah.
You take it, Mark. Because you have the transplant experience.
Yeah. It depends. I mean, so there's a number of things, and we were very invested in this, and I went bankrupt with my previous company and had to restart it with my own investment because we were very interested in using exosomes. You're probably familiar that exosomes and microvesicles are intercellular communicators, and so it's very clear, and we've shown this multiple times, that full-length mitochondrial DNA exists in exosomes. So we were... Yeah, but anyhow, so what happened is we were trying to get exosomes. We were then going to electroporate them and put in the full-length mitochondrial DNA and fuse it back and cure our patients. But what happened is I had a patient who showed up in my clinic who is a runner, but his mom died of MELAS 3243. And so he came to me and said, do I have it? And I did a muscle biopsy, and oh my goodness, it was 83% mutant heteroplasmy in his muscle. Yeah. But the guy had a good VO2 max. He ran a marathon in 3:02, which is a good time. So anyhow, he said he was tired. We did a blood sample, and he had leukemia. So he had a bone marrow transplant. And we were right in the middle of our experiments. We were getting the CHO cells growing in culture. We were all jazzed up. Got some great stuff in vitro. And then I said, wait a sec. He is a bioreactor inside his bone marrow with a tracer. So what we did is we measured his blood, and sure enough, it was full of the mitochondrial haplogroup of the donor. His exosomes were full of beautiful mitochondrial, normal, non-MELAS mitochondrial DNA. And so we muscle biopsied him. We did, with ethics approval, of course, urine sediment, skin biopsy, how many molecules of the donor mitochondrial DNA went into his muscle, or indeed, 3 of the 4 tissues in the human body, 0, not 1. So after being bathed in beautiful exosomes full of full-length mitochondrial DNA, not 1 molecule went into any of the 3 or 4 tissues in the human body. So that, it gives me some pause for concern. Mm-hmm. The issue then is, yes, there is evidence that, because again, bone marrow is where most of our circulating fragments of mitochondria come from. So then if there were mitochondria floating around, why did it not help? Why did those new mitochondria not help to repair? So 2 years of being bathed in mitochondrial DNA and with circulating mitochondria, none of them shifted into the cells. So yes, I know there's good experiments.
Why would you think it would?
What's that?
Why would you think it would? It's beyond me as to how you'd expect external mitochondria to get inside the cell.
Well, how did they get there in the first place? I mean, that's how they came to.
You make them there.
And that's where this whole, the research clearly.
It as a therapy.
Absolutely, there's clearly evidence that if you have damaged
well, there's this
heart,
mitochondrial transfer story, but I mean, that they're talking about for curing
right, but
disease,
that's coming from the blood into the tissue, and there is evidence that with the damaged heart, people are showing some transmission. But I don't think for our patients it's going to be able to replace in a non-injured tissue, all of the dysfunctional mitochondria based on our experiments. That was published in FAST Journal a few years ago.
All right, so let's go back to the question we asked before, ACEs, mitochondrial function, and what to do about it.
Yeah, this is a tremendously exploding area of research. ACEs, adverse childhood experiences, have been studied in a health context for decades now. So first they were studied in terms of finding that people who report more than 3 adverse childhood events, so that would be having, being exposed to abuse or emotional deprivation, or having a parent with an addiction, witnessing domestic violence, et cetera, is related to more disease in your lifetime, earlier disease, early mortality. It's been related to earlier cell aging, shorter telomeres, a more accelerated epigenetic clock, higher inflammation. But now we're talking about transgenerational transmission. So ACEs in a mom so her early childhood experience is associated with the baby coming out on day 1 of life with shorter telomeres, with accelerated epigenetic aging. And there are many different mechanisms. So one is acquired during the prenatal development. So this would be related to the developmental origins of health that you were mentioning last night, where the experience of the environment of the baby as it develops, the uterus, is under stress. And it changes everything, right? So the baby's developing in accord to the expected environment it's going to live in. If it's going to live in a threatening environment, it needs to have certain things like a methylated glucocorticoid receptor, so it responds to stress in a certain way. So for telomeres, we know that there are, we believe there's a development effect from a mom experiencing stress during pregnancy that affects how the baby comes out. That's been shown with lots of studies, including Janet Wojcicki here at UCSF. But also...
a collaborator of ours.
Right. And there's also a direct epigenetic transmission. So parents with a genetic disorder for short telomeres have offspring who tend to have short telomeres even if they didn't inherit the genetic disorder. And that's because the germline, the sperm and egg, carry the short telomere. And so that's just a direct transmission to the egg or through the egg. So
what do you do about it?
yeah, so we also have done interventions during pregnancy. And I will say that we have remarkable effects from pregnancy interventions on health of mom, health of offspring. I can tell you about those. But we also know that the health of the gametes, the sperm and egg, the epigenetic marks, the cellular health is shaped before pregnancy. And so we want to target preconception health as well. And because pregnancy is so hard to predict, most are unplanned, it's almost impossible to do a study on preconception health and then follow people and see if it helps them. So we just need to do it as a public health initiative. But in our pregnancy, many pregnancy studies have found that mindfulness and stress reduction and good nutrition, these all have important effects on development of the baby's metabolic health as well as emotional temperament. And we've, of course, studied the biomarkers as well. And in our study with mindful eating, mindful stress reduction, we found that these high-risk moms who tend to be low income and overweight tend to have, if they get some stress reduction during pregnancy, it was like an 8-week class, they have improved metabolic health. They have lower glucose on the glucose tolerance test, lower levels of impaired glucose intolerance, and
no change in body weight,
right,
but improved metabolic functioning.
Right. And less depression, and we follow them with my colleague Nikki Bush, and 8 years later, the moms still have less depression. It's a crazy effect, but the bottom line is this is a very plastic, critical period. Estrogen makes the brain extremely, I'll say, prone to neuroplasticity and modeling. So any intervention is going to take and have long-term effects in a positive way, as well as negative. So in terms of the negative effects of ACEs, of course, it affects brain development and emotional responding, and that can have a kind of a long arc or a shadow or footprint lifelong. So this developmental biology, both in the womb and early childhood, when the child's still developing, we really see pretty prolonged and dramatic effects of stress. Partly in the telomere area, it's not just affecting the circulating cells, but probably the stem cells. Every cell is bathed in the same blood, and the same stress biomarkers are affecting them.
In autism, there's a balanced nutrient supplement called Spectrum Needs that is commonly used.
What's it called?
The Spectrum Needs. There's about 30 different mitochondrial cofactors, and that actually help to fill in holes that might exist in the metabolism of a child, with, in that case, autism. But I'm wondering, this is a question for Mark. Do you have a Trim 7 version for kids?
No, we don't.
Well, I think, the other Mark, never mind.
That's all right. I think he was talking Mark Tarnopolsky. We got 2 Marks here.
No, we don't. Obviously, we haven't tested safety in children, but from a pregnancy perspective, the 2 number 1 reasons for infertility are obesity and advanced ovarian age. By the time a woman's 35, the ovarian function has gone down dramatically, which is strongly related to mitochondrial dysfunction. So we essentially used our mitochondrial core and a similar version of the Trim 7, but we pulled out the mint extract, and we showed that in the obese animals, we had 0 copulatory success. So if we'd have wild type as our control, and you have X number of pups born, and then you have obese animals, we had 0 copulatory success. With the animals treated with the supplement to improve mitochondrial function, we restored to 70% of wild type. And we had a bit of a screw up because there was a delay coming across the border. And our aging studies, that was published, our aging one came out 2 weeks ago. And our mice were so old that we had 0 copulatory success in the older mice because they were too old. That was restored to 20% with the supplement. And in the middle-aged mice, which represents approximately a female age of around 32 to 35, we doubled fertility success in those animals. So it does seem to have an effect on 2 known metrics to lower fertility, namely obesity and aging, both of which affect the mitochondria. One final question, just to follow up on the lovely discussion that you were having about prenatal exposure. So we know from World War II that with forced starvation with the Nazis, that the children born to moms who underwent starvation were very thrifty. And the methylation changes, the epigenetic changes, were profound, but it makes so much evolutionary sense, right? If you are in an environment where you have restricted energy intake, you want your offspring to be thrifty, which is great during evolution when there is no food. But in this, massively food-filled environment, it promotes obesity. And other interesting changes, Alison Holloway has done some work with nicotine, showing that if you expose a mouse to nicotine, the F2 generation, so 2 generations downstream, the grandchildren of those mice still had mitochondrial dysfunction and a propensity towards obesity. But it could be partially reversed by exercise training. So methylation is great because you can adapt quickly without waiting for evolutionary changes to occur to adapt to your environment. So you can not only induce the changes, you can reverse them too, at least the exercise.
Well, the reverse is the hard part. It's not that easy. On that subject, you're coming, I swear. 10 seconds. There was a paper that came out in Nature in December showing that babies born in Britain before 1953, when sugar rationing was lifted, had a lower incidence of diabetes and hypertension than babies who have been born after 1953. So the presumption is that those babies weren't starved. They were just exposed to less sugar, and they did better. So another reason for mitochondrial dysfunction again. Now, thank you.
Yes.
Go ahead.
Thank you very much. Thank you all for your nice and fun presentations. I have a lot of questions for each one of you, but maybe in another occasion. So for now, on a little bit different topic, I would like to know your point of view, your thoughts on genetic engineering to improve our mitochondrial DNA. For instance, relocating it, protecting it from ROS.
Ooh.
Genetic engineering?
Yeah, genetic engineering of mitochondrial DNA.
In
CRISPR
a human.
would do nuclear DNA. I don't know how you do. Well, how do you do mitochondrial?
So what we are proposing and what we are researching is moving the mitochondrial DNA to the nucleus, recoding the mitochondrial DNA so it can be read, transcribed, and translated from the nucleus, so then the proteins can integrate into the OXPHOS chain.
What technology are you using?
It's called allotopic expression. It's still in development.
Right. I think the problem with any genetic manipulation is delivery. How are you going to get CRISPR-Cas9? How are you, other than adenovirus, which has limited issues, it's really hard to get any. I mean, everything works in vitro, but trying to do it in a human is really a challenge. I mean, delivering CRISPR-Cas9 in blood to a person, where's it going to go? It's not going to get anywhere. It's a real challenge. And
I'd say Eric Schoen has actually done a lot of this work on ATPase6 involved in the NARP mutation, because all the intrinsic mitochondrial proteins are so hydrophobic, it turns out that without the right chaperones, getting it to the location, many of them, it's very difficult. But having said that, there are some great papers that Eric Schoen at Columbia has.
By the way, all those chaperones are very ATP intensive, so keep that in mind. You got a question?
Hi there. Great talks today. Kunj Patel. I'm a local physician that does some aging research. I was just wondering if you all had some thoughts or comments on the known mitochondrial inhibitors of complex 1 that people take commonly, things like berberine or metformin, combined with or without exercise, and if you think there's any utility or harms that you can foresee. I know definitive trials haven't been done, but you're making your own products, so I was just curious about that. Yeah.
Mark Mattson, why don't you go ahead and take that?
Well, so this is a kind of a normal situation where there's evidence from cell culture and rodent studies that imposing mild mitochondrial stress on cells can be beneficial. and in models of disease, often beneficial. I mentioned DNP and mitochondrial uncoupler is actually a company that is doing clinical trials with low doses of DNP in certain neurological disorders now. So in theory, it might be possible because things like that they distribute pretty well when you give them systemically. We did some work with 2-deoxyglucose, essentially restricting calorie intake at the cellular level. And we found in the short term it could be neuroprotective, but then Don Ingram and the group, he put it in the feed of animals and did a lifespan study and actually shortened life. And then there's a question of dose and timing. They were fed ad libitum, one dose. So again, this intermittent dosing thing is very important. Anything that's going to induce stress, whether it's mitochondrial stress or restricting energy availability, it's got to be transient so that there's a recovery period. And this is an issue for clinical trials. I think those kinds of things are worth trying once you've established safety.
Tom, go ahead.
Yeah, hi. Our Epel and Lustig in a K-12 curriculum from Coast, the Skinny on Obesity, talked about the danger of hyperinsulinemia on prospective mothers, not let alone pregnant mothers. Yet my county is very diligent on prenatal ATOD warnings. How much of the boat are they missing by not talking about what we learned in the Skinny on Obesity? What's the magnitude of what they're turning their backs on, if any?
Tom, what would you want them to be saying? He's an expert too.
I would want my county health department to listen to my Coast professors and not miss the boat on the dangers of hyperinsulinemia pre-pregnancy.
The question is, what is hyperinsulinemia? And the answer is, hyperinsulinemia is mitochondrial dysfunction because insulin is the proximate, downstream result of when your mitochondria don't work right. When your mitochondria work right, you're shunting energy through mitochondria to ATP and carbon dioxide. You don't need any insulin. You only need insulin if you have to divert, because you have to basically tell the cell to store, to export and store, and that's the job of insulin. So fasting insulin is the single best marker for mitochondrial dysfunction, and it's dirt cheap. The problem is that the American Diabetes Association says don't draw a fasting insulin. Now, why do they say that? They say it for two reasons, both of which are specious, both of which are wrong. Here they are. Number one, they say fasting insulin levels are not standardized across platforms. Right. That is true. Okay? Different assays give you different results. I agree with that. So what? If it's high, you got a problem. And if you use it as a baseline, you can then use it as a biomonitor for any given patient as you try to improve their metabolic status. So if their fasting insulin goes down, as long as they're on the same platform, you can make head or tail of it. That's the first problem. Second problem, they say fasting insulin levels do not correlate with obesity. That's exactly right. They correlate with metabolic health. We've already learned today that there's subcutaneous fat and there's visceral and there's even liver fat. The fact is, subcutaneous fat is where your body wants to put extra energy, and it's actually beneficial to do so. The patients who don't have subcutaneous fat, we call lipodystrophy, they are the sickest people on the planet and have the highest insulin levels because they don't have any place to put it. So, in fact, both reasons that the American Diabetes Association promulgate for not drawing fasting insulin are wrong, and to me, that is the biggest problem we have. And you go into your doctor right now and you say, I'd like a fasting insulin level, and they say, number one, you're not going to get reimbursed for it. Number two, I don't know how to interpret it. Number three, you don't need it. Okay? That has to change. And as far as I'm concerned, that's my job, is to fix that. And I'm dedicated to fixing that. But I got a whole lot of doctors to reach.
I'll help.
Yeah, I'd just say to step back earlier, right? So the poor mental health and stress happen before disease gets dysregulated enough that anyone would measure in clinical markers. So biosensing and measuring and educating young children, et cetera, about perceived stress is going to be really incredible prevention. And in terms of biomarkers to measure, I do think that I agree with Rob that we really missed the boat on that. And
we sure did.
Yeah.
Please.
Hi. Actually, this is for Bob.
By the way, anybody else with questions, it's now or never, because we're almost at 5
Especially
o'clock.
women. I'd love to see some women speak up to balance out the almost mantle.
Do what I can. Can you just talk, for Bob, talk a little bit about, your view of mitochondrial adaptation rather than dysfunction? Because when we talk about dysfunction, I think we can forget that a lot of that we have to look at the context of what the mitochondrial is doing, and the contextual thing. It's not like when you have one that's broken because of genetics, but most of us have pretty normal mitochondria. So, can you just clarify that?
Bob, that's really for you. Yeah, I mean, there's functional mitochondrial dysfunction, and then there's, more like genetic mitochondrial dysfunction. Yeah.
Yeah, so I usually think of this as abnormal persistence of an otherwise good thing that's necessary for healing. So when a fragmented mitochondrial phenotype persists, it leads to chronic inflammation. And on the other spectrum, and actually the brains that we've looked at in Parkinson and some adults with autism, there's been a hyperfusion phenotype in mitochondria that goes the other direction. So for me, it's a matter of trying to unblock the programmatic changes that are necessary to complete those steps of healing. And that's where I think at the moment we have a number of supportive things that people have talked about, but I think that there'll be a whole new class of medicines that will help start that transformation.
I'll listen. I just wanted to clarify, because I was just thinking that most of the people that we see who have, quote unquote, they've got all the signs of metabolic illness or whatever, but it's just usually because there's some source of inflammation, whether it be dietary or something else going on, and that's what's creating this mitochondrial dysfunction, if you will. But there's nothing. I'm just trying to get the point that Bob has made many times.
Wait. You made an assumption. You made an assumption that the inflammation caused the mitochondrial dysfunction. Yeah. The mitochondrial dysfunction caused the inflammation.
Yeah, that's right.
Well, that's where.
Right?
Yes.
Right?
Well,
right.
Okay. Well, no. But,
well, it is bidirectional.
No. See, that's where. In my worldview, underneath the reason the mitochondria are reacting like they're in danger, like there's inflammation, is because there's something bothering them.
Oh, yeah.
In the environment.
For sure.
Yeah.
Absolutely. There's all sorts of substrates that lead to mitochondrial dysfunction. But one of the throw-offs of mitochondrial dysfunction is ROSs, and ROSs activate NF-kappa B directly. So there's your
right, but to me, that's part of the process.
Gateway to inflammation.
That's what I'm. To me, I see that as part of the process, not as something. I mean, that's helping you protect yourself. Except you should turn that on and turn it off. You're not turning it off. You're in trouble.
Yeah.
So maybe it's just semantics.
But all right, Elissa, come. Your turn.
Hi, thanks so much. I'm a pediatrician, and in the first 1,000 days of life, we know how important the microbiome is in modulating epigenetic outcomes, reducing risks of atopy or mental health concerns later on. So when we're talking about these diet and lifestyle interventions that positively impact the mitochondria, they also positively impact the microbiome. Mind-body medicine positively impacts the microbiome. Sure. All of these obesogens, endocrine disruptors, mitotoxic and microbiome disrupting. So how do you tease out? I always start with the microbiome, right? I also have a lot of kids with mitochondrial dysfunction, but why not marry the two, because there's such microbiome mitochondrial crosstalk?
Because we don't have any microbiome experts on the dais, that's why.
Well, we do know that microbiome diversity is very important. And there's a number of bacteria which are associated with metabolic dysfunction. Butyrate seems to be positive, so bacteria that release butyrate are associated with better mitochondrial function systemically. So there's clearly a feedback. I mean, there's trillions and trillions and trillions of bacteria there. And a couple of things that do influence this, clearly exercise. We've published, others have published, that exercise has a favorable effect on the microbiome. And so enhancing diversity is important. And that really gets back also to what we eat. So highly purified and highly processed sugars are not good for your microbiome. Eating closer to nature, nuts, a variety of other things really help to improve the biodiversity of our microbiome, which then feeds forward and then feeds back likely to the microbiome. So it's very complicated and still an emerging science, but I agree it's really important. Interestingly, one of the number one things we found in beta testing when we started giving this TRiM7 is 100% of people had a significant change in their gut. So we are going to be looking at microbiome because we suspect 10, 20, 30, 40, 50% of the effect is probably the favorable effects on the microbiome. And we think that's probably how it's working.
I got one word, and you know it: fiber.
Oh, yeah.
I knew it.
Fiber.
That's right. Well, I'd love to hear from
Depends
Dr.
on the
Epel,
fiber.
because one of the studies that looked at improving heart rate variability in children and how that could improve microbiome diversity independent of diet. And so, and on the other hand, on babies 0 to 3, we want very little diversity, almost all bifidobacteria, right? And so are there ways that we could, I mean, I just would love a look at younger children. I mean, younger children are really excluded from so much of the research, and this is where we need to start to really prevent all of these problems later on.
Yeah, no argument there. All right, last question.
Thanks so much for all the great talks. This is kind of a more clinical question because I work at Kaiser's MECFS clinic. And one of the big questions now is the rapamycin as a treatment. There recently was a study, actually in humans, not great, I understand, but I guess, is there any other reasons? I was wondering what you guys thought about it.
Does the term decerebrate ring? I think it's the craziest idea in the world. I mean, number one, what happens to older adults? They become relatively immunocompromised. Then you give them a drug which affects their ability to respond to inflammation. But more importantly, with older adults, it's not, and this is coming from me, an endurance athlete, it's not just the mitochondria and increased VO2, it's strength. And Blake Rasmussen, training with Bob Wolf, has 2 publications clearly showing that rapamycin completely suppresses the ability to respond to exercise and amino acids. So people are going to be sarcopenic. It's going to amplify sarcopenia. So yes, they get their great little results on one side. But again, day, night, winter, summer, we have to cycle through periods of stress and recovery. And that's going to completely blunt it if you take rapamycin. It's,
the goal here is not to de-stress. The goal here is to harness stress. Okay? And rapamycin basically undoes that phenomenon. So I don't see that as, there are a lot of people trying to use it. I think they're all going to fail.
And they already are.
Okay. I understand this is in normal people, right? Like I'm saying
we're talking about normal
MECFS.
people.
I'm talking about MECFS, where they have shown that like a large percentage of people actually felt better.
Sorry.
Oh, I'm sorry, chronic fatigue syndrome.
Which?
Chronic fatigue syndrome.
Oh, chronic fatigue syndrome. Bob, you study that.
But if you're going to age, you don't want to lose your muscle, so I don't see any rationale. And if you can tell me what causes chronic fatigue syndrome, and that would be great. I know Bob has some thoughts on it. But
I'm not a fan of it for MECFS.
Okay.
Or anything.
All
Except
right.
organ rejection when you get an organ transplant.
All right. Well, first of all, I want to thank you all for staying.
Stay tuned for the 2nd Annual Symposium on Mitochondrial Health, Healthspan, and Aging.